So, from here on out, the chapter gets quite long and convoluted, and I would say probably as convoluted as the nephron tubule itself. So what I want to do is hit on a lot of the high points, because if you look through the chapter, you'll see there will be page after page and diagram after diagram that all look pretty much the same. So I have pulled out the slides that I think are the most relevant and added some of my own illustrations. So let's talk about what the purpose of the kidney is to filter out the bad stuff. So we want to see how well our kidney functions by looking at the glomerular filtration rate. And so this is the amount of filtrate that the kidneys produce each minute. And it should be about an average of 125 milliliters per minute, which is about 10% of the fluid coming from the blood that's being delivered to the kidneys. So about 10% that's leaving the bloodstream and entering into the capsular space in Bowman's capsule. So we can estimate this by looking at the amount of creatinine. So creatinine is a toxin that's produced from the breakdown of creatine phosphate. And typically the kidneys are very good at getting rid of it. And none of it is resorbed back through the tubular system. So almost none of it is resorbed. So what ends up happening is a good estimation for kidney function and glomerular filtration rate, because if you find a lot of it in blood serum, that means that the filtration rate has gone down because we're not able to clear the creatinine. So there are other tests that are better than the creatinine test. One uses inulin. But for most purposes, if you go in to check your glomerular filtration, they'll probably use a creatinine clearance test. All right, so now let's see why filtration happens. So we're going to look at the filtration pressure. So to do that, let's go back to our glomerulus and our capsule, and let's look at the filtration membrane. So the glomerulus, as you recall, is a fenestrated network of capillaries. So the endothelial lining has these pores in it, and then that is going to be bound to a dense layer. And then you have your podocytes, where there are pedicels, or their foot processes, that are going to make these filtration slits. And this is going to create our filter system. And so we're going to have a couple of things that's going to drive the movement of fluid and particles from the blood vessel into the capsular space, one of which is the hydrostatic pressure. And that is just simply the pressure that the blood exerts on the capillary walls. And that's going to be due to the fact that we have blood flowing and it's under pressure. And the afferent arteriole, as we've said, can be one of the things that regulates that pressure, because if we dilate the afferent arteriole, we're going to allow more blood in there and we're going to have more pressure. So this is a hydrostatic pressure of blood. And of course, if we constrict the efferent arteriole, we limit the exit of blood here. So what that's going to do is cause a backup of blood in the capillaries, and that would, in effect, increase, again, our hydrostatic pressure. So that's one way to increase hydrostatic pressure. We could decrease hydrostatic pressure by, for example, constricting the afferent arteriole, and that would reduce the amount of blood that comes into the glomerulus, so it's not as under high pressure. By the same token, we can open the efferent arteriole up, and that will allow more blood to flow out, and once again, the exit rate will be higher, so we can reduce the pressure within the glomerulus. And this will be part of some of our auto-regulation methods that we'll talk about a little bit later. But first, let's look at some of the forces at work here. So we know we have our blood hydrostatic pressure. We're also going to have our colloid osmotic pressure. And these are, as you recall, the things that are suspended in solution, so our solute. So when we look at blood, it has the plasma proteins. And plasma proteins cannot get past this filtration barrier. So basically, we're going to have plasma proteins stay behind, and other solutes can cross. Small ions, organic substrates, things like glucose, vitamins, amino acids, and so forth. They can cross. But the albumins, the majority of them cannot. So we're going to have a colloid osmotic pressure by the fact that we have these large proteins over here, but not over here. All right, so when we look at it, we can look at our glomerular hydrostatic pressure, and this is typically the pressure just of blood exerting force against the capillary walls, and this is about 50 millimeters of mercury. Now remember, we are also going to have the hydrostatic pressure within the capsule because we're going to have some pressure of this filtrate that comes in here, and it can only leave via the proximal convoluted tubuli. So, there's going to be some limit of how fast it can leave. So there is going to be a capsular hydrostatic pressure as well, and it's only about 15 millimeters of mercury. We also have our blood colloid osmotic pressure. This is going to draw blood out of the filtrate and back into the capillary, because while we have a lot of solutes leaving, we still have those plasma proteins left behind. And so some of that fluid will be reclaimed because it's going to go back towards where there's the higher solute concentration. So that will be our plasma proteins. So really, when we look at it, by the end of it, the solute to fluid levels will be balanced by the time we get through with this process. But more on that in a minute. So what we're going to look at right now is the net filtration pressure, which is going to be all three of these different things combined. So our blood colloid osmotic pressure, drawing fluid back into the capillaries, which is going to be opposed by our blood hydrostatic pressure, which is pushing fluid and solute out into the capsular space. And our capsular hydrostatic pressure is, now that we have fluid in the capsule, it exerts its own pressure, but it's much, much less than the blood hydrostatic pressure. So basically, if we subtract the capsular hydrostatic pressure from the blood hydrostatic pressure, we end up getting about 35. So when we have our blood colloid osmotic pressure, which also tends to pull fluid into the capillary, we have an outward pressure of 35, an inward pressure of 25. So our net filtration pressure is going to be about 10 millimeters of mercury. And this is fairly typical at the glomerulus. One thing to note is that the capsular colloid osmotic pressure is pretty much zero, because we're not going to have plasma proteins in the capsular space. Now, if you have a pathology, then sometimes they'll get in there, and let's say the filtration membrane is damaged for some reason. Then some of these plasma proteins can get in here, and then that will skew the balance, and basically we'll have some colloid osmotic pressure within the capsule. So in a healthy kidney, we can just say it's negligible and say it's zero. All right, now let's talk about something else that's very important. Let's talk about osmolarity. And osmolarity are the total number of solute particles per liter. You can say per unit volume. So if you want, you can look at milliosmoles per liter or milliosmoles per milliliter or so forth. So at any rate, it's typically the total number of solute particles per liter. And this is different from osmolality, which is per kilogram, and I just want to make that clear. But for most medical purposes, we deal with osmolarities because it's much easier to withdraw solutions and measure the volume than try to figure out the kilograms of, or the weight of, the actual solutions. Now, the one thing about osmolarity you have to be aware of is that you take a volume of stuff and you heat that volume, the volume will expand. So in this case, you can see how the osmolarity would change if you heated your solution. But typically in the body, we don't have to worry about that. The things in the body are going to stay about the same. We're not going to have great expansions and contractions of volumes within the body. So when we measure stuff, we usually just draw the blood, measure it, and it'll be measured in osmoles or milliosmoles per liter. Now, body fluids typically have an osmotic concentration of about 300 milliosmoles per liter. One other thing that I should emphasize, and I know this is a review, but it's something that's important. So we're talking about number of solute particles per liter. So if you had 1 mole of sodium chloride and stuck it in 1 liter, you would have an osmolarity of 2. Why? Because the sodium and the chlorine dissociate. And so now you will have 2 moles, a mole of sodium and a mole of chloride ions, dissolved in 1 liter. Now, if you had calcium chloride, which, since calcium is a divalent cation. It would have 2 chlorides associated with it. If you threw that into a liter of water and let it dissolve, you would have an osmolarity of 3, because now there are 3 particles. There's 1 calcium and 2 chloride ions. So this is something to be aware of. But in any case, the take-home message is that the renal physiology is going to be dependent of changes in the osmolarities of the fluids as they move through the renal tubules. And also, when we look into the renal medulla itself, especially when we looked at the juxtamedullary nephrons, we will see that the renal medulla, the pyramids within the renal medulla, are going to have increasing concentrations as we go deeper of solute particles, so their osmolarities will increase. And this will be very important, and this will be some of the driving factors of what we're going to see when we look at important terms that we're going to talk about in just a minute, such as countercurrent multiplication and countercurrent exchange. Before I get into that, I just wanted to review very quickly what osmolarity is. All right, so here's something that I drew on the board just as a quick recap. And what I wanted to do before I got into a lot of the big, points that we're going to get into is I wanted to highlight the major processes within the nephron. And so obviously my drawing, just like all the others, is going to be folding out that nephron so that you can see it. And typically our distal convoluted tubuli over here would actually be interfaced here with our afferent and efferent arteriole at the juxtaglomerular complex. But for illustration purposes, obviously we sort of pulled it apart so we can take a look at what happens in each portion. So I wanted to go over some of the major things. Obviously the filtration all happens at the glomerulus, and then we'll have our filtrate leaving in the proximal convoluted tubuli. It will go through here next. And then in the proximal convoluted tubuli, we're going to have resorption of most of our salt and our water that entered the filtrate. So these are valuable things. We're also going to have filtration of other ions, or I should say, sorry, not filtration, resorption of other ions, calcium, magnesium, and so forth, and almost all of our organic substrates, so amino acids, vitamins, glucose, and so forth. So this is going to happen at the proximal convoluted tubuli. So 60 to 70% of our sodium chloride, 60 to 70% of our water will be resorbed here. Now, once we enter the loop of Henle, we will see that the descending limb here will be freely permeable to water, but not to solutes. So we will have about 25% resorption of water by osmosis from this descending limb of the loop of Henle. Now, as we turn and go to the ascending limb, especially when we get to the thick part of the ascending limb or the thick ascending limb, we're going to have the active pumping of sodium chloride out of the thick ascending limb and into the interstitial space. And that is going to set up something called the countercurrent multiplication. Because what's going to happen is as the filtrate enters into the thin limb, or the descending limb here, the thin descending limb, we're going to lose a lot of water. And that's going to concentrate the solute in here. And we're going to have some salt left over. So as we turn, we will see that we really have a very high concentration of solute. So up here, we started out about 300 milliosmoles per liter. When we come down here, we'll gradually descend through 900 milliosmoles down to about 1200 milliosmoles. So we got a really high concentration of solute down here. Now, as we turn and ascend, then we got all these sodium pumps that start actively pumping out the sodium chloride. So salt's going to get out of the filtrate and it's going to be put into the interstitial fluid. So we lose about 20 to 25% of our salt that we started out with in the filtrate. So that's going to be pumped into the interstitial fluid. And here, where the concentration of salt is the highest, the pumps are going to be most active. And so we're going to remove a lot of our salt down here in the deep part of the medulla. So as we go up, we're going to pump out less and less to the point that we have reached about 100 milliosmoles up here. So what's going to happen is the more salt we pump out, the more osmosis of water we have on this side. So the more salt we pump out on this side, the thick ascending limb, the more resorption of water we're going to have on this side. So basically, what we're doing is we're setting up a countercurrent multiplication. What do we mean by that? The descending limb filtrate is going in one direction, the ascending limb filtrate is going in the other direction, and the more salt we pump out on this end is going to pull out the more water on this end. So it is a positive feedback mechanism, and it's a multiplication effect because the amount of water we absorb here is going to be dependent on the amount of salt we pump out here. Now, as we get up here, we're going to adjust the amount of water and the amount of salt. It's variable. It's going to be dependent on basically our filtrate volumes and our fluid balance within the body. So at the distal convoluted tubuli and the collecting duct, we will see that we're going to have variable water resorption under the influence of antidiuretic hormone. Remember our posterior pituitary hormone, antidiuretic hormone that is also known as vasopressin. And what that does at the kidney is it causes these aquaporins, which are these water channels, to be inserted into the tubular cells so that now water can pass out of the filtrate into the interstitial space here. So basically water can leave. When aquaporins are inserted in here, then water can leave. And aldosterone causes these aquaporins, which are normally sequestered within the cell and not necessarily active. Once they get put into the cell membrane, now the water has a free passage. And the same is true over here in the collecting duct. So we have aquaporins that will be inserted into the cell membrane under the influence of antidiuretic hormone, and that will allow more water to be resorbed. So it's a small amount, it's maybe 5% of the water we start out with over here. And then we also have variable resorption of sodium chloride under our adrenal hormone, aldosterone. So remember, this is an adrenal cortical hormone, and this is going to cause us to pump sodium chloride out. So we have sodium pumps that are going to be activated when aldosterone is present. So we have the aldosterone. So these things, as we say, work hand in hand. Now, another thing I wanted to point out is called something called countercurrent exchange, and that is going to involve the vasa recta. And one thing I wanted to point out is when we speak of the vasa recta, or the vasa recta, however you want to say it, these are those straight capillaries that are going to follow the loop of Henle. Now, this is basically pertaining to our juxtamedullary nephrons. So our cortical neurons just have their peritubular capillaries. Now remember, our peritubular capillaries are going to follow along in the proximal and distal convoluted tubules, but when they dip and make that long, straight vessel that follows the loop of Henle in the juxtamedullary neurons, we call this the vasa recta. So all of this is the vasa recta, and I've just sort of put it out here in cartoon form just to show you how the fluid in the vasa recta, the blood flow, is going to be going opposite that of the tubular fluid flow. So this is going to be our countercurrent exchange. So we're going to have stuff that was pumped out here be picked up by the capillaries. And as we go over on this side, we're going to have quite a bit of solute that we picked up over here that's in the interstitial fluid, and that solute is going to cause a lot of resorption of water over here. So basically, the actions of the vasa recta will exactly balance that of the leaving of sodium and water over here within the loop of Henle. So this is going to be important because that's going to maintain our concentrations of salts and waters in the interstitial fluid. So we're going to have a very nice salty concentration in here, and the vasa recta is going to help maintain those concentrations. Now, I put this table from the book here, table 26.4, which is on the next slide. There it is, 26.4, because this sort of is a good table that summarizes everything going on in here. All right, now, we've already talked about the osmolarity, and I wanted to touch on this again because the next thing we're going to look at in some detail is this picture. And this summarizes why the countercurrent exchange works and why countercurrent multiplication works. So when we have our filtrate, it's about 300 milliosmoles per liter, and it will start moving through the proximal convoluted tubuli. Now, in the renal cortex, it's about 300 milliosmoles per liter in the interstitial fluid as well. But as we descend, the filtrate descends in the loop of Henle, what's going to happen is we're going to have resorption of water. Now, that's going to concentrate the solute in here to about 600 milliosmoles. Down here, it's going to be about 900 milliosmoles, and so forth until we're about 1200 milliosmoles. So what's going to happen now? So we've got really a lot of solute concentration in here. So over here, we're going to have our sodium pumps that are going to be active, and they're going to be pumping out sodium chloride here. So as this very concentrated solution rounds the bend and our sodium pumps start becoming active, they're going to pump out solute, and they're going to pump it into the surrounding interstitial space. So the osmolarity of the interstitial space is pretty much going to match the osmolarity of the filtrate at each point. So the deeper we go into the renal medulla, the more salty it gets. And so that's going to be the driving force, because the more salt we get, the more solute we get being pumped out here is going to drive this osmosis, and it's going in all directions. They just show the countercurrent exchange here too. So our countercurrent multiplication is just simply the fact that the more salt we pump out, the more resorption of water we're going to get into the interstitial fluids. That water, of course, will eventually back up into the vasa recta. And so what we're going to see is that on this side, all this salt that's being pumped out in the interstitial fluid will be picked up by the descending vasa recta. And that's going counter to the thick ascending limb. Now, as we round the bend here, we've got a fairly high solute concentration here as well. And that's going to drive resorption of water from the interstitial fluids. That came about because we had all this water moving via osmosis out of the descending limb. And so that's going to be picked up by our ascending vasa recta and eventually carried back into our venous circulation. All right, so now we've got sort of a 7-step process. It very much mirrors these 7 steps shown here. So we've got our 7 steps here, and then we've got the slide form of it. So this just basically is a batch of word slides that reiterates everything that we've said. So we've got filtration of the glomerulus, and then we're going to have a lot of stuff going on at the proximal convoluted tubuli. We're going to get a lot of our ions and organic substrates back. We're going to get some water moving by osmosis out of the tubular fluid. And so this is going to reduce the volume of the filtrate. Keeps solutions inside and outside fairly isotonic. Notice they're isotonic all the way through. All right, now, until, of course, we get to the top. I should mention here, by the time we get up here, we're only at about 100 milliosmoles per liter in the filtrate, whereas the surrounding fluid will be about 300. Now, notice that the distal convoluted tubuli and the collecting duct, their permeabilities are going to be dependent upon our antidiuretic hormone and our aldosterone. So remember that a lot of their permeability is going to be under the regulation of hormones. All right, so once we get to the proximal convoluted tubuli, we reclaim most of what we want, the water and our organic substrates, our sugars, our amino acids, vitamins, and a lot of our ions. So the whole point of the descending and ascending limb of the loop of Henle is to concentrate the urine. So we're going to move from the peritubular fluid, water from the peritubular fluid into the surrounding interstitial space. So that's going to make the it's going to make the filtrate very, very concentrated. So remember, the tubular fluid is the filtrate, the peritubular fluid is the stuff in the interstitial space that will eventually get picked up by the vasa recta. All right, so this is going to reduce the water or the volume by water loss, i.e., so the water will be lost out of the filtrate by osmosis and be resorbed back into the interstitial fluids. Now, on the thick ascending limb, we've got this really, really high concentration of filtrate that once it starts to climb up, we're going to start having this active pumping of sodium and chloride out of the tubule. And remember, the ascending limb is impermeable to water. So that's the key. It's impermeable to water, so water can't follow the sodium and the chloride out. And we're also going to look a little bit later at urea. Urea is something that is a toxin that the kidney gets rid of, but it's also used for helping adjust the concentrations, the osmotic concentrations. We'll look at that a little bit later, but right now I want to concentrate on the countercurrent multiplication, which is what we're doing right here, and the countercurrent exchange, which will happen when the vasa recta picks up these things. All right, now, so in the distal convoluted tubuli and collecting ducts, we're going to have our final adjustment of the composition of the tubular fluid through aldosterone and through antidiuretic hormone mediated mechanisms. And so we're going to basically adjust the composition of urine through a bunch of processes, resorption, secretion, and so forth. We will also see at this area, we're going to be adjusting some of our bicarbonate ion and our hydrogen ion as well. Finally, in our distal collecting tubuli and our ducts, we're going to have, once again, our final volume of urine and osmotic concentrations of the tubular fluid. By the time we get into the collecting duct, the filtrate will be basically urine at this point. All right, so then we have the vasa recta. That's going to absorb our solutes that were pumped out by the ascending limb of the nephron loop, which is the fluids going in the opposite direction. And then it's going to help maintain this concentration gradient of the medulla. And then when the vasa recta ascends on the opposite side, it's going to be pretty heavily laden with solute. So all that water that was resorbed into the interstitial fluid will be picked back up by the vasa recta, and then that fluid content of that blood, by the time it leaves, will be fairly normal, like 300 milliosmoles, and it will then return to venous circulation. And so by this point, we have created urine. So by the time it gets to the collecting duct, it will enter the papillary duct, and from there, the urine composition really doesn't change. It will flow through a minor calyx into a major calyx, into the renal pelvis, and then down into the ureters to the bladder, where it will be stored until time for excretion. All right, so let's look at how we can control the glomerular filtration rate. We've already discussed some of these mechanisms, and they involve autoregulation. So this is what's going to go on in the local level. We also have hormonal regulation, which we will see with the release of renin by the juxtaglomerular complex. And then we have autonomic regulation. This sometimes goes hand in hand with hormonal regulation, but this is going to be by the sympathetic division of the autonomic nervous system. So let's start with autoregulation. So obviously we want to maintain our glomerular filtration rate. So there could be local blood pressure changes or blood flow changes. So we can change the diameters of afferent and efferent arterioles. We can also change the diameter of the glomerular capillaries. Remember those intraglomerular mesangial cells. They can help change those glomerular capillary diameters. So we can reduce or increase blood flow. So if we have reduced blood flow, then what will happen, or glomerular blood flow, we will dilate the afferent arteriole because we want to bring more blood in. We want to raise the pressure. We can dilate the glomerular capillaries and constrict the efferent arterioles. So we can force more blood into the glomerulus and then have more filtration. The other thing we could do is if we have a rise in blood pressure, so this is going to stretch the walls of the afferent arterioles, then this is going to cause the smooth muscles within those afferent arterioles to constrict. And so that's going to narrow the diameter of the afferent arterioles. That's going to decrease the glomerular blood flow. All right, so here's a picture of it. Here we have, in this case, a decreased glomerular blood flow. So we're going to decrease the filtration. So what are we going to do? dilate the afferent arterioles to let more blood in. We can contract some of the mesangial cells and constrict the efferent arterioles. That's going to bring our glomerular blood pressure back up, and then our homeostasis will be restored. Now, if we had the opposite effect and we had an increase in glomerular filtration rate, then we would basically dilate the efferent arterioles and constrict the afferent arterioles, and that would have the reverse effect. Now let's talk a little bit about the hormonal regulation of the glomerular filtration rate. So there are going to be 3 hormones that I'm going to talk about. The first of which the book doesn't really do anything but touch on, and that's adrenaline, also known as epinephrine, that is released from the adrenal medulla during generalized sympathetic stimulation. And what it's going to do, as you recall, the sympathetic system shifts function away from the digestive and urinary tracts and basically increases the circulation to and the metabolic activity of the muscles, so the skeletal muscle system. So what the effect of the adrenaline is going to do is it's going to reduce the glomerular filtration rate, because at this point we don't want to be thinking about micturating or urinating when we need to be thinking about fighting off the saber-toothed tiger. So basically what it's going to do is it's going to shut down both the afferent and efferent arterioles. So it's going to decrease the diameter of both of these vessels, and that's basically going to have the effect of just stopping blood flow or greatly reducing blood flow to the glomerulus. So basically, since our afferent arteriole, the diameter of it is our main regulator of how much blood gets in there, then by decreasing both arterioles, the afferent arteriole is going to have a bigger effect, and the net thing that we're going to see is reduction in the glomerular filtration process. But by the same token, basically what we're doing is we're just shunting blood flow away from the kidneys to other organs. Now, let's talk about the renin-angiotensin-aldosterone system and what it does. This is a little bit of a paradox because the effects are going to be different depending on the amount of renin released and the amount of angiotensin II that eventually is going to be in circulation. So this hormone, I should say, angiotensin II, also has receptors on the afferent and efferent arterioles. And the angiotensin II will cause the constriction of both of these. However, it has more of an effect on the efferent arteriole, so it's going to constrict it more. So at low levels of angiotensin II stimulation, we're going to have basically not as much constriction of the afferent arteriole, and we're going to have more constriction of the efferent arteriole. So what we're going to get is we're going to get more blood coming into the glomerulus, and therefore we're going to get a greater perfusion rate. So in this case, the angiotensin II will increase glomerular filtration. However, at very, very high levels of angiotensin II, we will see that both arterioles will be clamped down. So the afferent arteriole is clamped down as well, and that will have a very similar effect as adrenaline. It's going to reduce glomerular filtration. And think about it this way. If you have really, really low blood volumes, then you want to increase blood volume to increase blood pressure again. So therefore, you would want to restrict the amount of glomerular filtration so that you don't end up peeing away all of your important fluids. So the effects of angiotensin II on the glomerulus can vary depending on the blood volume. Now, the thing about angiotensin II is its whole job is to maintain the glomerular filtration rate, right? They're about 125 milliliters per minute. So that's why its effects can be variable. Now, let's look at the natriuretic peptides. As you recall, in the heart, we have the atria, which are very sensitive to stretching because they're going to be sensitive to the amount of blood volume because of venous return. So when we have a high blood volume in venous return, it's going to come into the atria, it's going to stretch them, and that's going to cause the atria to produce this atrial natriuretic peptide. And as the name implies, it basically is going to promote the excretion of sodium. And we're also going to have the ventricles also produce a natriuretic peptide called brain-derived natriuretic peptide, so called because it was first discovered in pig brain extracts, but it's produced in human ventricles. So brain-derived natriuretic peptide, breed ventricular natriuretic peptide, and atrial natriuretic peptide together will also stimulate an increase in glomerular filtration rate, because what we want to do is we want to increase the rate at which we excrete water. So basically, we want to pee faster, pee more, pee faster. And this will allow us to get rid of some of that excess plasma volume as we increase the glomerular filtration rate.